Now we come to a very important part of this afternoon. We want to give some ideas, some aspects, some points for coming in and for collaboration by having teaser presentations by the three chairs of the Austrian Microfluidics Initiative. They will be around the topics innovation in microfluidics, application potential, as well as teaching and education, and will be done by ERTL, Torsten, MAYR, and Günter LEPPERDINGER. These three people are the chairs from the Austrian Microfluidics Initiative, and we are very happy that we can now jump into the presentations. So thank you very much for you, Torsten and Günter, to have you both here. Hopefully Peter is tested negative at the end and can maybe join later. In that case, I would like to kindly hand over the floor to you, Torsten, for the second part of the presentations. Please.
Okay. Yeah, welcome to this to our teasers now from Peter, Günter, and me, and we want to give you some thoughts about microfluidics for the future. So, as I said, Peter ERTL is away; he can't take part. So I will take over his part. And you see, this is about technical innovations, and I think what is going on at the moment, we have already seen that in the presentation of George before, that there is this foil on chip, and these that really we can bring here different production technologies to this microfluidic area. And I think this is a big step forward, because then we can really have here higher volumes. And then I think the next step, there will be also some that microfluidic circuits will be have integrated functions. I think we have seen this also today already. What can happen, what is possible there, or what will be possible there in the future. And of course, there will be these circuits then also with integrated functions, what we have seen also already today with integrated sensors, integrated pumps. I think this is where the journey will go in the future, to really have this really smart devices with a high functionality. And I think this is also important, that there will be new functional materials with, of course, biocompatibility is a very important aspect, either when you work with cells or we work with blood samples. And there will be also a trend to have even smaller channels that require less liquid. And there will be, and we also have effective processes for the reagent dispensing, especially in this reel-to-reel technology. So these were the technology innovations, but there are also some scientific innovations, and there I think it will be going through precision medicine. In this case, where you have these really, you can use the chips and then make a screening for drugs that everybody gets his specific medicine. Of course, then there is also a lot of other integration here necessary, a lot of technology integration, which is illustrated here. And I think then we will end up in devices with microfluidic structures and a lot of other electronics with integrated circuits. And of course, in future, this will be all done with wireless communication or near field communication. So that then this is then bringing us more to digitalized a platform. So I think, yeah, and with that, I'm also want to point out more to the applications. What are application fields that we face in microfluidics? And to really get there, I got back to literature and make a literature search. And what you see here, this is I made this search yesterday, and the search term was microfluidic. And this is, I received about 73,000 documents with the term microfluidics. And okay, these are all research papers And I think a lot of them are also more with dealing with engineering and material science regarding the microfluidic chips. But I think at least half of it are really dealing with have a target application. And you see also the applications field are so manifold, and that's more what I wanted to show with that it's really application fields are from chemistry, so or biochemistry, what we have seen also today, and genetics, then also molecular biology, cell biology, all also in environmental science. Environmental science has also picked up microfluidics for detecting, for example, for detecting in oceanography, in marine science, to really detecting the nutrients like phosphate and nitrate. And this is then now done with microfluidics, and also many other agriculture and biological science. You see, it is really widespread applications of microfluidics, and it is an enabling technology. And a good example for that, what I have picked up now, and also because it's really important that in our times, is DNA sequencing. And I want to illustrate what this technology can really bring, or what the benefit of this technology can bring in. You see here, this is when the human genome has ended, and about 2001. And at this time, the cost for sequencing a human genome was $95 million. And you see here, over the time, the cost decreased to $700, and this was 2020. So you see, there's a huge technology jump has happened around here, and this is when the next generation sequencing technology was in place. And I think here it was really more and more applied, and when it was on the market. And you see how the costs were dropping, and you see also this would be the Moore's law if you just multiply and make it smaller. But there was a huge technology step in that sequencing technology, and part of this technology game changer was microfluidics. So you see here, this is what you have. What was the former sequencing technologies based on the Sanger method, and you see here most of it will take place in microtitle plates, or this dedoxy method, and all these reagents were placed in microtitle plates. And then you had this capillary electrophoresis, where you then analyze it with a spectrogram. So it was multiplexed, but it was quite time effort. There was a lot of time effort, but this was then really reduced with a lot with this next generation sequencing technologies. There are a few of them, but the most popular is the Illumina. They have a market share at the moment of 90%, and what they use is a microfluidic flow cell at the core of their system. And this here you see that this is this flow cell, and in this flow cell are these cavities here, these micro-sized cavities, and here takes all the biochemistry magic place in these cavities. And really to bring the liquid to that, you had these flow cells. And I think this is a really nice example how microfluidic chip is an enabling technology, despite all the biochemical chemistry progress that have been made to establish this technique. Then I'll give another example, and this is an AMI collaboration between Peter ERTL's group and my group, and we realized nanotoxicology measurements in microfluidics with integrated sensors. And there we used oxygen and pH sensors, which then are for monitoring of cell cultures. So here you see on the bottom of this, you have these adherent cells, and then we expose these cells to nanoparticles, and then we can measure the cell metabolism. We can measure how these nanoparticles really influence the cell metabolism by measuring the respiration, and the acidification of the extracellular matrix. So this was done with these, you see here, this is the microfluidic chip, and we realized that, and this is something I also want to stress here, and that is what we also do, or what we see as expertise in the army, is that we have these, that we made these, the whole process from the design studies to mass production. So we started with these design studies by drawings and by first prototypes in the labs. And then we already went to a rapid prototype, new rapid prototyping method that this was established together with the project partner KDG, which is a injection molding company in Austria here, and they made from these rapid prototypes with machines that they formerly used for DVD processing. And you see here, these are, and we can really realize very fast these prototypes. And then we made from this, we made this first microfluidic chips with integrated sensors, also with methods that can be used in a mass process. And we ended up with a final injection molded chip in a very, very short time frame. So, and this is also something what we had the application in mind, and we really to show what is necessary to bring this in a reasonable time from the idea to the final chip that you really can mass produce. And the last thing I want to also explain is I think we already heard a lot about organ chip today. I don't want to go into detail on that, on what organ on chip is. I think probably all of you know that, and I have given here the definition of the organ on chip society, which I also encourage you, if you're interested in that, to become a member. And this is then about what this former organization of this organ on chip society, it was in the ORCIDE, called the ORCIDE project, where scientists and also persons from the regulators and also from academia and also from the institute were collected this roadmap and developed this roadmap, and they illustrate here what is necessary to get really to application of your technology. And see, there was this at the beginning. There is this initial development made by academia and also by organ on chip company, but to bring this really to the applications where this roadmap has identified several steps. So there's they need specifications to really make ready to use organ on chip devices, and also qualifications. So these independent tested centers have to be set up really to give good quality of results, and there must be also a lot of standardization. I think we all aware of that everybody uses different connectors, different materials, different media. So there's a lot to do in this standardization, that we really get to a point where we have ready to use organ on chip devices. And then it is also then production upscaling. And if we have solved all these, then we have there's ready for adoption by the industry or by academia, and then we can maybe serve all these applications like pharma and food and cosmetic. But I think this is picked that up this illustration because I wanted because it's a good example what is necessary to really bring a lab result to the application. And you see also the time scale here. Yes, and okay, I have also, and with that, I think I will hand over to Günter, and he will give you some insights about what is necessary for, or what he is thinking, teaching and education in microfluidics, where are the problems.
Thanks, Torsten. I'll just give you kind of a hint to move on with the slides, so that I just go briefly through that. So thanks for invitation. Thanks for being with us. Also, thanks for the interest on the AMI initiative. So to be very, very brief and give you kind of a short, conclusive information on what interests us being at the university and being responsible for education and training of young scientists. I have to add to this really nice talks and impressions we got from your side, which is mostly research and development centric. That at the universities, our prime aspect is, and I'll just want to stress and emphasize on that, is education training of young people. Albeit there is a lot of research also going on. You know, this is, I would call it a mass production right now. And due to the fact that most of the young people coming today to us and are willing to be trained in a very hard discipline, namely biosciences, I guess this is obviously also driven by Corona. So there's also positive effects of Corona. But being more precise on what we are talking today, the point is what we often try to neglect is that although there's applications, technology, and maybe also this precise analytical topic, we certainly neglect or ignore what it needs in order to handle all the data, and also in order to handle personalized privacy and safety problems around the patient or the individual to be tested. So I just want to stress that first thing, because we have this feeling, and it's a deep feeling, that although we make progress in analytics and point of care, the idea here is that you give something into hands of people who might not be trained. This is one part. And although they could be trained, they are not willing to actually try to find a surrounding where privacy and safety is really secured. And for this, we need to also make sure that people who are trained and educated have an open mindset, and therefore, you know, to this rubric, which you find on the right side, analysis and engineering and biosystem integration, and also this kind of hard and software problem, the data analysis management needs to be really enforced and maybe also improved in a way that we can tackle these important questions. Others, the point will remain that this, and this will be on the next slide. No, sorry. You see on the left corner that a chip in a lab might not be able to change or be replaced by this interesting idea, lab on a chip, which can be converted into a point of care device and point of need device. So these problems is something that I guess in the academic world needs to be a future topic and a perspective which we need to give utmost care. So on the next slide, I just want to introduce you to another big problem in training and education. What we see in academia is that in most cases, on the left column, you would have attributes that appear to be common to us. In case you're working in industry, you would see the other side of the coin. And if you take this together, it's a double-edged sword. So in case in academia, we're working on prototypic problems, the industry wants us to be innovative. So marketable, commerciable, everything should be in a way driven by thoughts that are actually applicable to the market. So in case we train people, we often try to be at the spot in the lab where single scientists work on one chip, which, as we do not have a lot of money, should be reusable. And in case, and we saw that today, the idea here is that it should be high impact. The idea in the industry actually is completely opposite. It's high throughput, it should be disposable so that you can earn on replacing the not worn out, but the just used chips, and so on and so forth. Now, if the same problem applies not to the technology, but to the basic research and the approaches within the field, the idea in academia now persists, and we see that also from the funding point, that it should be transdisciplinary. In the industry, maybe people just in case of transdisciplinary would go for a B2B model, but at least this is my experience. The focus is extremely narrow. And at the other end of this kind of list, you would also see that the innovation, which they start out as a basic idea, should be kept secret. So now we have this dichotomy, which we have to solve also for the problem of teaching in academic setting, and maybe also in research and development. And therefore, my question, at least my personal question, is what should we actually care for in terms of education and training? Should it be the biomicrofluidics device engineer, which obviously is around and can do a lot of things, because we've seen that today, and it's very, very impressive for me always to see what can be achieved and what the accomplishments at the degree of sophistication are. It could be also that we should just educate the analytics design manager, or what is thought in many aspects, that we should go for high throughput. Torsten has approached that. Or at least what I think is very, very important, that we also need to care for data evaluation and compilation. With that said, thank you for your attention, and again, thank you for this kind of initiative by organizing this extremely nice webinar.